So class, now that we've had a brief overview of bacterial genetics and DNA replication, let's have a crash course and a super brief overview on transcription and translation. Oftentimes this will be referred to as protein synthesis or the central dogma of biochemistry. Let's define our terms. Transcription, as this word implies, if we look at the root word, is going to be copying information down. So during transcription, we copy information down from the form of DNA to the form of RNA. These are both forms of nucleic acids. So we'll have this information encoded in the language of nucleic acids as we copy it down from one kind of nucleic acid to another kind of nucleic acid. The next is translation. During translation, we're going to take that information that's stored in nucleic acids, particularly ribonucleic acids, and then we'll use that to make a protein. So we'll switch chemical languages. We switch from the language of nucleic acids to the language of amino acids when we go from the RNA to the protein, hence the name or the term translation for this process. And this was the central dogma. This is what was very commonly understood and taught and thought, and then we started to find the exceptions to the pattern. We have RNA-based viruses that can take their RNA, particularly their antisense RNA, convert it to sense RNA, so the negatively stranded to the positively stranded RNA. Or we can have retroviruses which will take their RNA and convert their RNA to DNA, and then their DNA will go back to RNA, and so on and so forth. There are also a wide variety of regulatory RNAs that have been discovered and studied in the last 15 years. Smaller interfering RNAs is a classic example. RNA interference is a pretty hot topic right now in molecular biology. So here we have our base chunk of DNA. We'll take that piece of DNA and transcribe it. We'll copy the nucleic acid information down into other nucleic acids. That could be transfer RNA, that could be messenger RNA, so tRNA or mRNA, or we could have rRNA, also known as ribosomal RNA. Of those three kinds of RNA, messenger RNA is the simplest, ribosomal RNA is the most complicated. So we go from DNA to RNA during transcription. Then during translation, we take that messenger RNA and feed it through a ribosome. And that ribosome will read the information on the messenger RNA and use it to pull in transfer RNAs or tRNAs that then start transferring amino acids to the growing protein. And keep in mind that ribosomes are made of rRNA and other protein molecules. And at the end of translation, we have your favorite protein molecule. Working off to the side, we have our regulatory RNAs, our smaller interfering RNAs, our microRNAs, our antisense RNAs, and those will either activate or deactivate genes, or they'll activate or deactivate translation. To look at this in more detail, you can see that for the transcription phase, we first need to unzip the DNA. We unzip that DNA using DNA helicase, and then we have a special enzyme that will make a polymer of RNA, that messenger RNA. We call that enzyme RNA polymerase, and there's different flavors, different varieties of RNA polymerase. What I want you to know is that we first unzip the DNA, and then we use the information on the template strand of the DNA as a template to code for a messenger RNA molecule. And we'll have a start sequence and an end sequence on our template strand of DNA. After we go through transcription, we are going to have the DNA be continually unzipped on the leading end and re-zipped on the lagging end. So our direction of transcription is going to be from left to right on the screen, and the messenger RNA molecule continues to grow in length, and we'll continue to unzip the DNA on the right side and re-zip the DNA on the left side until we hit that red line, the termination sequence, which tells us to stop translating our DNA to messenger RNA. During transcription and translation, we're going to follow standard base pairing rules. A is to T's, B and G is to C's. Something that should be noted, though, is that when we go from DNA to messenger to RNA, there are not going to be any U's. There will not be any uracil present in that DNA, or the messenger RNA. So as I look up here, I can see that there's some T's present and I can see some T's present here as well. So this may be a typo. I need to double check with our publisher on that one. Here is an animation that is included that I thought was fantastic. The RNA polymerase will unzip the DNA and bring in messenger RNA polymers. Those messenger RNA polymers will then attach to the growing strand of RNA, and then that RNA will move out of the nucleus and will have the next step take place. In bacterial chromosomes, we can have multiple RNA polymerases going through multiple rounds of transcription at the same time for the same gene, making multiple pieces of messenger RNA. Messenger RNA, and all RNA, all ribonucleic acid, is very similar to DNA or deoxyribonucleic in terms of the general structure, with two major caveats. The two major caveats is that RNA, generally speaking, is going to be single-stranded, and generally speaking, DNA is double-stranded. The second big caveat is that, generally speaking, there are no thymines in RNA. Instead, we have U for uracil in RNA. RNA can form secondary and tertiary molecules depending on the complexity of the sequence. This is through RNA base pairing with itself, and then we'll have things like hairpin loops that will form. Another major difference between ribonucleic acid and deoxyribonucleic acid is the 5-carbon sugar involved. RNA contains a ribose sugar, DNA contains a deoxyribose sugar. Other RNAs include microRNA, antisense RNA, riboswitches, and small interfering RNAs. These regulatory RNAs are not as well understood as the mRNA, tRNA, and rRNAs. They're the new kids on the block, so to speak. We aren't going to spend a lot of time focusing on these regulatory RNAs. Primer RNAs are active in both bacterial and eukaryotic cells. These primer chunks of RNA are used to initiate RNA synthesis, much like a primer chunk of DNA is used to initiate DNA replication. A ribozyme, as its name implies, is an enzyme that's made out of RNA. And this is kind of the exception to the rule. When I ask a student, what is an enzyme made out of? The answer I'm looking for is protein. Enzymes are made of proteins. Every now and again, though, the minority or subset here is enzymes that are made out of RNA. These ribozymes are going to remove other unneeded sequences from other RNAs and have other regulatory functions within the cell. The classic example of the ribozyme is going to be in the ribosome. These ribosomes are made of rRNA, some of which act as an enzyme, which speed up a chemical reaction. The chemical reaction in question here is forming a covalent bond between adjacent amino acids to link them together. To review again, ribosomes are made of ribosomal RNA, and there's two primary subunits to the ribosomes. We have the big subunit and the small subunit. The ribosomes between prokaryotes and eukaryotes are different, and this will be very significant in a couple chapters when we talk about how antibiotics work. The differences in ribosomes between a prokaryotic organism or a bacterial organism and a eukaryotic organism like you and me means that we have some targets for our antibiotic drugs that hopefully will have a very strong effect on the bacteria and a weaker effect on the patient. But I'm not going down that rabbit trail now. We're going to focus on DNA synthesis. So in our ribosome, we will have that messenger RNA molecule get sandwiched between the large and small subunits. Within that messenger RNA molecule, we have little chunks of nucleic acids. These little chunks of nucleic acids are going to be arranged in sequences of 3. Every 3 nucleic acids is known as a codon. I'll say that again. In a messenger RNA molecule, every 3 nucleic acids is known as a codon. That's very significant. The frame of reference for these codons is going to be noted by the AUG, the start sequence. On the transfer RNA, we are going to have an anticodon that matches up with a codon in the messenger RNA. So if I had an AUG codon in my messenger RNA, the anticodon that matched up with it would have to have a UAC to match up with that. A UAC anticodon would match up with an AUG codon, according to base pairing rules. A's goes to U goes to A. Remember, we're in RNA right now, not DNA. In DNA, A matches with T, but in RNA, A matches with U. So A goes to U goes to A, and then G goes to C. We have a UAC anticodon that matches up with the AUG start codon. On the other end of our transfer codon, or transfer RNA, or tRNA, opposite from the anticodon, we'll have an amino acid attached. Depending on the specific tRNA, we are going to have specific amino acids that correspond to the specific anticodons and codons that are coded for by the messenger RNA. The ribosomes of the prokaryotes, as I've emphasized already, are different in size compared to a eukaryote. So they have different total sizes and different subunit sizes. Our small subunit's always going to bind to the 5' end of the messenger RNA. Remember, DNA has directionality. DNA is read from the 5' to 3' direction. And that directionality in the DNA gets transferred to the messenger RNA as well. Messenger RNA will also have a directionality that is the mirror opposite of its template strand. And that messenger RNA is also going to be read in the 5' to 3' direction. After the small subunit binds to the messenger RNA, the small subunit being the small subunit of the ribosome, the large subunit of that ribosome is going to bind, and then that large subunit will start to synthesize a protein molecule by catalyzing the chemical reaction between adjacent amino acids, forming covalent bonds to link those amino acids together. There's a special name for the covalent bond that holds adjacent amino acids together. That special name is a peptide bond. A peptide bond is a covalent bond that holds two adjacent amino acids together. Here we can see this process in action. We're going to have 3 sites in our ribosome. We have the E, P, and A sites. We have the exit site, the peptide bond formation site, and then the access site. A for access, P for peptide bond, E for exit. So we will have a transfer RNA enter the access site. If the anticodon on the transfer RNA matches up with the codon in the messenger RNA, then it will stay put. It will form hydrogen bonds and stick in there. If the anticodon and codon do not match up, then the transfer RNA bounces back out and a new one bounces back in. And this process is a random process. Random tRNAs will bounce in and out of the access site until one bounces in that matches up. And that one that bounces in that matches its anticodon to the codon is going to have a specific amino acid that's going to be the next one in the protein. In the P site, we are going to have the amino acid in the P site attached to the polypeptide chain that's forming and exiting from our ribosome. And then finally at the end, we are going to have the empty transfer RNA leave from the ribosome. It will drop off its amino acid. In this case, we have a triangular purple and a rectangular purple amino acids. Those are dropped off by the transfer RNAs, and then the transfer RNA will leave empty-handed without an amino acid attached. When we look at these codons in the messenger RNA, those groups of 3 nucleotides, they're super important. They dictate which specific amino acids are going to be added to the growing polypeptide chain. Because there are 4 different nucleotides, and there are 3 base nucleotides total in an amino acid, we have 3 to the 4th power. So 3 times 3 times 3. 3 times 4 times 4 times 4. Oh my gosh, I'm having such a brain hiccup right now. I'm grabbing a calculator. I'm so sorry for this brain hiccup. We have 4 times 4 times 4. There we go. We have 4th to the 3rd power. I promise you, I'm not an idiot, although I feel like an idiot right now. So because we have 4 nucleic acids, A's, T's, C's, and G's, we'll take 4 to the third power. That third power corresponds to the 3 nucleotides that are present in our codon. Those 64 different codons that are possible will code for the 20 available amino acids. Because we have more codons than amino acids, there's going to be some overlap. Some amino acids are going to have multiple codons representing them. There's some redundancy here, and this redundancy allows us to have the correct amino acid still inserted into the growing protein, even if we've had a mistake in DNA synthesis or in transcription or during translation. This redundancy helps to minimize the effect that a mutation has on the cell. As our transfer RNAs are bringing our amino acids into the ribosome, there's going to be a little bit of wobble. This wobble means that the first 2 nucleic acids in the codon and the anticodon are the 2 most important ones. They are absolutely essential for getting the correct amino acids. The third amino acid doesn't necessarily need to base pair perfectly with the adjacent amino acid. This means that this redundancy that we have allows for a mutation that occurs to be not necessarily have a catastrophic effect on the protein, and allows for us to have a little bit of variation without a change in the end product. This wobble is a good thing. It helps to minimize the effect that mistakes have as those mistakes appear in our genetic information or in our genome. Here's a codon cheat sheet. I never, ever want a student to memorize the codon cheat sheet. If I ask you to do transcription and translation on the test, which I'm telling you right now, I will have you do. I will always have at least one transcription and translation question. You will need to be able to use this codon cheat sheet to tell me what amino acids are going to be in the protein. So if I look at our codon cheat sheet, let's say I had the AUG to start codon. The first codon is A, the second codon is U, and then the third codon, the first nucleic acid is A, second nucleic acid is U, third nucleic acid is G. So to find the AUG codon, I would start with A in the A row, go to the U column, and then find AUG. AUG codes for our methionine, our start codon. If we look at leucine right here, all codons that code for leucine have a C and U for the first 2 nucleotides. Any codon that starts with a C and a U will always code for leucine. This is part of that wobble that I was talking about. If I have a C and a U, that gives me a leucine. It doesn't matter what the third nucleic acid is in that codon. Or another good example is proline. Any codon that starts with a CC is always going to code for a protein, for a proline, regardless of what that third nucleic acid is in the codon sequence. So here's a wonderful summary slide. We have in our DNA triplets. A triplet is a segment of 3 DNA nucleotides. That DNA triplet on the coding strand is going to base pair with the DNA triplet on the template strand. That DNA triplet on the template strand is used to directly determine the messenger RNA. So the T in the template strand goes to A in the messenger RNA, A goes to U, C goes to G. So now we have our start codon. And that AUG in the messenger RNA, that codon in the messenger RNA, will base pair with a UAC anticodon on the transfer RNA. And this transfer RNA will specifically carry a methionine amino acid into the growing polypeptide chain. And because we have different DNA sequences in our DNA triplets, we'll have different codons, and those different codons correspond to different anticodons, which means we get different amino acids. So let's look at this codon right here, ACU. According to this figure, ACU codes for a threonine amino acid. But let's double check. Let's go to our cheat sheet. We have the A row, the C column, so we'll go over and down, ACU codes for threonine. When you are studying up, make sure that you are able to look at the codons within a messenger RNA molecule and figure out which amino acids those correspond to. When we have a bacterial piece of messenger RNA being translated, we are going to be able to have multiple ribosomes bind to that piece of messenger RNA at the same time. And it's very efficient in the bacterial cell. This can happen. We can start translation before transcription is even finished. So here we have transcription occurring, and that piece of messenger RNA is being spit out. And then once that piece of messenger RNA, once that strand of messenger RNA is sticking out, we can have a ribosome bind to it immediately. And that ribosome will continue to follow that replication fork as we are producing a piece of messenger RNA. Something that I need to emphasize on this slide is that this AUG, this start codon, gives us our frame of reference. When you are trying to go through a piece of messenger RNA and find out what your frame of reference is, the AUG versus UGC versus GCU, that different frame of reference is going to have a very big impact in what amino acids end up in your protein. And if you change the amino acids of the protein, you change the structure or the shape of the protein, and then you change the function of the protein. So this is a big deal during protein synthesis. Getting the right frame of reference, finding that AUG sequence, and then pairing off every 3 nucleic acids after you find your first AUG is going to help you get the correct amino acids in your protein sequence. Another important thing is that the correct frame reference here will help you find the correct stop codon. There are 3 codons that tell you to stop: UAA, UAG, and UGA. If we had the wrong frame of reference here, we could accidentally hit a stop codon prematurely. Here is a summary figure from your textbook showing translation. This translation process is going to involve first the messenger RNA being fed through our ribosome. During this process, the ribosomes will have transfer RNAs bring the correct amino acids in by base pairing the codons to the anticodons. Then the peptide bond will form between the 2 adjacent amino acids. Those 2 adjacent amino acids are then going to start to be ejected out of the top of the ribosome, and the empty transfer RNA will also be ejected from the ribosome. The ribosome then scoots along the messenger RNA molecule. A new transfer RNA is brought in that brings in a new amino acid. This new amino acid forms a new peptide bond, and then we have an empty amino acid ejected from the exit site. The access site is now empty again, and a 4th amino acid is brought in that matches up its anticodon on the tRNA to the codon on the messenger RNA. That 4th amino acid will form a 3rd peptide bond, and this process continues, so on and so forth. In bacterial cells and eukaryotic cells, start codons are always going to be AUG. AUG is the only codon that I'm requiring you to memorize. All the other codons you can use the codon cheat sheet for, but you should memorize AUG is the start codon. It's worth mentioning or emphasizing that messenger RNA can code for several genes in a series in bacterial cells. We'll get to those later. It's called an operon. But in a eukaryotic cell, one strand of messenger RNA will always code for 1 protein molecule. It's always a 1-to-1 ratio in eukaryotes. But in bacterial cells, one piece of messenger RNA can code for multiple proteins. And these multiple proteins, generally speaking, are all going to be related to each other in a metabolic pathway. In a bacterial cell, since there is no nucleus to separate transcription from translation, transcription and translation occur at the same time or simultaneously in the cytoplasm, whereas in eukaryotes, transcription occurs in the nucleus and then translation occurs in the cytoplasm. In a bacterial cell, the genes are pretty simple. We're going to have an uninterrupted set of triplets coding for our genes. But in a eukaryotic cell, that's not good enough. It has to be more complicated than that. So after we go through transcription, after we copy down our messenger RNA in a eukaryotic cell, we need to edit the messenger RNA. And during this messenger RNA editing, we have segments that we leave in and segments that we remove out. The segments that we remove are called introns, and the segments that we leave in are called the exons. Yes, those names are backwards. I completely understand that it's totally counterintuitive. It should be the other way around, but introns are removed and exons stay inside the messenger RNA molecule. Here in a eukaryotic cell, we can have our DNA template transcribed. Here's our unedited piece of primary messenger RNA, and it has exons and introns. And the exons will stay in, the introns are removed. So here we can have the intron removal shown. And then we have only exons left over in that messenger RNA molecule that's now done being processed. That messenger RNA molecule in this eukaryotic cell, now that it's done being edited and processed, can leave the nucleus and enter into the cytoplasm so that translation can occur, so that protein synthesis can occur. So, concept check class. Which of the following kinds of RNA or ribonucleic acid contains hairpin loops and is involved in carrying amino acids to the ribosome during the translation process? So what kind of RNA carries amino acids into the ribosome? Is it messenger RNA, regulatory RNA, ribosomal RNA, transfer RNA, or primer RNA? If you do not know the answer, you can pause this video, flip back in your notes, or you can rewind the video, or you can check your textbook. 5, 4, 3, 2, 1. The correct answer is D, transfer RNA, or tRNA, as it is also referred to as. If you have any questions about protein synthesis, please post them on the discussion board or shoot me an email. Happy studies!